Hello. Hey fam. Hey fam. We live. We live. How's everyone's morning going? Hope we got some people in here to hear about plant tissue culture. So, let me just give you guys a little time. Welcome everybody, welcome. We're going to talk about plant tissue culture. Give everybody just a little bit of time. Alright. So right now we're talking about plant tissue culture, and I wanted to get started with just showing you guys what the end result of plant tissue culture looks like. So, let me try and prop you up. There you go. Hey! We made it. Here we go. So, this is Sequoia dendron giganteum, and this is giant sequoia. And so, when you look in this, you can see that this was coming from a cutting. And so basically what happened was you either can start off by seed, which is really awesome because the genetic properties are strong. It's not like cloning, so like if you guys have heard from cloning, some of the genetic material can actually be damaged through cloning. So it's not actually like the best way to propagate and sustainably grow more plants from another plant. So what this happened was, what happened here was, so this is an Agarose Gel, and so if any of you guys do like gel electrophoresis, black bacteria plating, and things like that, you're probably familiar with agarose, but I'll touch on it a little bit. So agarose comes from seaweed, and it's just like, you know, an organic gel that in most cases you can add sucrose to, and it will allow for whatever your medium to be, to metabolize. And so a lot of the times when you make a good media, you will see like at the bottom right here, it's starting to metabolize. This dude is really actually like 3 months old, which is a trip. Because, yeah, I mean, it's a sequoia, it's a giant sequoia, so it hasn't grown extremely, I don't know, not a lot of exponential growth. But so, I just wanted to get into the steps of plant tissue culture so that if anyone really wants to cultivate some plants themselves, you're totally welcome to. It has to be an aseptic environment, so if you have a laminar hood or just a ton of isopropyl, you'll be fine. I actually like to wear a carbon mask also when I'm performing tissue culture, just because, you know, when we talk, we're exhaling a lot of bacteria. So let's actually work with this plant. This plant is native. This is our milkweed. By the way, I'm in California. Hey, welcome everyone. Shout out to where you are. So we'll use this guy. This is a native plant, really common. This is our mustard plant. I don't know, maybe I said Asclepias, milkweed. Been working with that a lot, but no, this is the mustard plant. And so basically what would happen is you start off by cleaning your plant. So what happens is this is where all the genetic material goes down. It's between all of these internodes. And so if you see right here, you'll be cutting aseptically with a scalpel. You will cut right here and you'll cut right here. And this, let's actually cut it to show you an example of what really it would look like. This is actually just an extra organic matter. You are going to want to cut this off because this is actually going to cause more energy expansion. So we don't need that part of the plant at all. Cut that off. So here you have it. I wish I had some forceps, but definitely not at the lab, so I'm working with what I got. But let me turn around this camera if that's, yeah, I can. And so we'll go over this again. We have our sequoia, and that is a cutting from the tree, so it's not from seed. And so we have our plant, and when we make our cutting, what we're shooting for is between, I'll actually use this. Do you see how this is like the main part of the plant? And so what we're going to shoot for is cutting above and below this growth right here. And so what that means is that's a part of the plant where a lot of genetic information, nutrition, and food, the sucrose in the water is taken up. And so when we cut right there, what we're allowing for is the optimal nutrient uptake. And so we would actually end up with something like this. And so this is the internode. And what we would do is if I was in the lab, I'm at home, happy Mother's Day everyone, we'd actually insert this into our Agarose Gel, and that would be going from the bottom up. And so it's going to end up looking like this. So you can practice this at home. It's super fun. You can actually follow my friend Test Tube Gardens. He sells kits online where you can actually propagate on your own. I really suggest that if you're interested in doing any type of like plant tissue culture. It's super fun. And so we can also start with just talking about when you get started with your plants. A few things about plant tissue culture: you're always going to want to start taking a 5 to 7 inch cutting from the vegetative plant, and you cut off as many nodes as possible, which is this part of the plant we're talking about. So we're going to remove all these large leaves, and then we transfer this cutting. It's going to be a bunch of little pieces. We're transferring that into a 15% bleach solution with preferably deionized water, but if you don't have deionized water, that's fine too. You can also boil it, and so you want to have your magnetic stirrer going, and you just have it in that solution stirring for about 5 minutes. Once you're done with that, you'll empty it out, rinse it out with DI. I always go with DI because especially in this kind of aseptic environment, like you just don't want anything with high ppms. It just could mess stuff up. So what you're going to use is we actually go with different medias. Each of our media has a ppm, also not parts per million. It's a plant preservative media. And so what that is doing is that's actually helping with the oxygenation of the plant cells. So what's going to happen is they can oxidize really quickly when they're actually in one of these small little tubes. And then you'll have your 3 grams of agarose. You want 7.5 grams of your sugar. You can buy a sucrose from like Thermocycler or whatever, or any other big science companies, but we go to the local grocery store and buy the off-brand sugar. It works perfectly fine. Food dye is really important just because it's easier to identify, like what mediums, what hormones have you added to your media. And so at that point you want to know whether you want to root or shoot, and you can add like charcoal. Activated charcoal you can add. That's really good. And that will actually help show the seed that it is ready to be germinated. It actually takes away the light. So at that point, the light won't get to the seed and you can actually grow. It thinks it's still in soil, which is really cool. Let me see what else we can talk about for the tissue culture. It's kind of hard because I'm not in the lab right now. Performing tissue culture like this, there's different types of media also in terms of the plants that you want to use. So there's WPM, which is a woody plant media. This is what this media is. And so that's like anything that's a tree, anything that's like woody, hardy, anything thicker than a shrub. There's also going to be Myrskagi and shug, which is a type of mushroom, I believe. And what that's being used for is that helps with more flowering plants, the more soft, like more bendable plants. The rule I use is if you can bend it, then you would want to use the Muskogean shoot, which is MSS media. But yeah, you can also do the rooting media, which has a lot of auxin in it. And so that's just a plant hormone that promotes growth, and you can add that to your media. And so at that point, it'd be really cool because you have your activated charcoal in your media, and that's allowing for everything to go dark. So the seed is thinking that it's submerged into the soil, and then on top of that, you have all of the sucrose allowing for it's photosynthesizing, helping with photosynthesis essentially. And so then after that, you would be able to add your plant hormone, and once you add that, that would actually tell the plant whether it wants to root, shoot, and that depends on what you're starting with also. So you could be starting with cuttings, you could be starting with seed. That all depends on what you want to work with also. There's also different pHs that you would want to set. So essentially, with the pH, I believe, if I remember correctly, the pH is about 5.8, generally speaking. So you can use, ascorbic acid, really. You can use anything simple that's going to help bring the pH up or down. Also, with the containers that you want to use, it's really important. You just want to make sure that you're using like a borosilicate or anything that's like Nalgene, or I think there's another one that's, I forgot what brand, but it's basically something that you can autoclave. It's going to withstand large amounts of heat, and you want it also, once again, to be super aseptic. So, oh yeah, I forgot to mention, right now I'm not currently working in a plant tissue laboratory, super sad. I'm doing bacteria and mold culturing, which is fun. But, so yeah, I haven't been with this in a while, but still really fun, still really interesting. The thing about using tissue culture with plants that I think is really important is because at this point, this is a really sustainable practice, you know. I encourage anyone that if you're gardening, if you're working outside where you're throwing away a large amount of plant material, remember that you can tissue culture all of this material. So sometimes it's amazing to me, but you'll see a large mulch pile or something that someone's going to throw out, and you're like, wow, you can make a lot of cuttings from that, and you can actually regenerate so much of the plant and seed itself. And I think that that is such a sustainable approach right now, because we're going through a mass extinction when it comes to our biodiversity. So that's really upsetting. If you guys have any questions, please let me know. Oh, waiting. I also have my lab book from the plant science days over here too. Hey, welcome. I don't see you yet, but welcome. Hello. Hi. How's it going? Good, good. Thanks for joining in. Cool. So, are you working with plant tissue culture or anything similar to that, or? Oh, so cool. Yeah, I'm a student too. That's really awesome. Well, I'm working on it, but took a little break to work in a lab, but I'm going back. But welcome, welcome. Do you have any questions about tissue culture? I'm from US, sitting here in sunny California, talking about some plant tissue culture. I'm just trying to let people know that it's pretty easy to do it at home if you have an aseptic environment. It's pretty important in a sustainable sense also, I think. Kind of see what everyone else is up to also. Yeah, pleasure talking with you too. What are some of the most rare but effective media you have used? Rare but effective. Um, that's a really good question. I think that I'm not sure if I've used like rare media in particular. I mean, media is so different in general to where, like, right now, I work with uh, scanning materials for pathogenic, gram-negative mold and yeast growth on agarose plates. So it's like media can be used. Oh, you can even use media for gel electrophoresis. So in that sense, you're not actually culturing anything, but it's media. So the media is so broad. But I think the coolest media might be for me the Mirskagi and Shug, cause it's like a cool name. I thought that was pretty cool. I want to say it's like derivative of a mushroom. I'm not positive though. Uh, I understand that the people that make it, I think, are from Japan. Um, but I also think that some of the interesting aspects of the media, I'm hoping that like this is kind of answering your question, is the role that we play when, for example, we add like activated charcoal to the media and we add a seed, and then the seed is convinced at that point with the right type of hormones that I'm in the soil and this is the perfect condition to start growing. And then, it pops up and ends up being in a tube, which is pretty cool. And so this is also interesting. So when you're growing in a test tube, this is actually fantastic because it hasn't reached the point of contamination. So this was done in a super aseptic environment. It was done correctly. I've had this for like months. Shout out to Test Tube Gardens once again. But yeah, so that's another thing with the agarose. If it's high quality, it's not going to have any colonies or any type of growth growing on it. And the plant should just be taken from the agarose. Effective media is usually within the pH of 6. I think it's 6 for plants. If it's bacteria, your pH is actually, you're going to start becoming more alkaline. You're going to end up with a 7.3. That's what you want for bacteria. So it's actually pretty different. And at that point, the media has to be, it's just a matter of being aseptic because media, it can pick up anything. I mean, there's this one epidermis bacteria that grows, and it's a single skin cell. It grows on McConkey, which is another type of media. And McConkey, we color that red. And that will pick up literally skin cells. Like, that's another crazy media. Kind of veering away from plant tissue culture media and talking more about bacteria media. But media is just amazing because it's weird, yo. So our mouth is like a petri dish, like for real. Our body has I don't know, like a radiant heat setting of like, 98 degrees on average. So that is really like an incubation chamber. And then I don't know if you've ever read your container. So when you have like your agarose powder before you start to weigh it out and add DI, the agarose powder usually says like beef bovine or like cow pancreas, things such of that fun sort. And it's interesting about media because that is really, it's trying to act as if it's our body. I mean, it's a living organism. What we're doing is we're trying to we're trying to make a condition that would replicate something growing within our own body, which is crazy to me because that's media. That's, I guess, the rarest media. I don't know about that, but that's pretty funky. I think that it's cool that essentially our mouths, I mean, if you guys ever have time and some of you guys work in a lab and you guys are doing bacteria culture, swab your mouth and do some gram stains on it, or like do some like gel electrophoresis. Find out, run a DNA ladder and be like, what is in my mouth? Because it's pretty crazy. Our the yeast that grows in your mouth overnight, that's the same just as in a petri dish, an agarose petri dish. Just pretty crazy. So talking more about the media, I just want to bring up that troubleshooting with media often can involve sometimes the hormone is not right. So like for auxin, if you don't add enough auxin, or the plant preservative media is also really important, because at this point, when you're growing for so long, you're going to pick up environmental contaminants, and you're going to end up with all sorts of stuff on the plate. Another really random fun thing to do with agarose is you can do environmental testing. You can do it at your house even, which is really cool. If anyone, also you guys have access to buy agarose online with DI, or you have some from your lab, straight up take some agarose, and it doesn't have to have crazy plant hormone or anything in it. This type of agarose, I actually don't know the acronym to this one because I'm still learning, but it's called a TSA plus. And TSA plus is basically, it sets at a 7.3 pH, and you can open the plate in your house and let it set for exactly 30 minutes. And within 30 minutes, you can put it in somewhere that's dark, damp, and sitting at about, I think it's like 50 to 60 degrees. Like, it doesn't even have to be that hot, really. And in a week, go check your plate. It might be stanky, but you probably have some growth, and you can see environmentally, like if you have a gram stain kit on hand, you could totally check that out and see like environmentally what's up, what's going on, because that's pretty crazy, man. Media is just so cool in that sense that there's so many different applications for it. It comes from a plant. It comes from seaweed mostly. The agarose, I mean, for like other media, like I said, it's like beef, bovine, and pancreas, cow pancreas, just like really funky things. But yeah, let me see if there's any other questions. Yeah, if any of you guys have questions, feel free to ask. What do I think of? Let me just try and think of some questions. Hello, Morali. Good morning. Hope you're doing well. I think also that when performing tissue culture, you should definitely mess around with it and work with culturing the seed, the plant itself. I mean, you don't have to have like a laminar hood. It would help, but just work and see what you got. And the most important thing is it's like sustainable. I would like to see all the plastic one day be made by like a silicate, like a plant-derived silicate, because I know if you guys have ever worked in like an aseptic lab, all of the plastic that you go through, it's insane. Like, I probably go through around 200 plastic syringes a day for inoculation. Not even using like the inoculation loops when you swab or when you streak a plate, but just like in general, just grabbing the material and placing it onto the agarose. There's a lot that's in waste. So it'd be really cool that if somehow tissue culture was involved in propagating large amounts of renewable silicates, such as like hemp or soy, or I think there's certain mushrooms also that they were using that they can use the cell wall and it can be renewable. So I think that most importantly, tissue culture could be a huge step in sustainability in our planet and helping lessen the use of petroleum, plastic, all of that nasty stuff like polyurethane and things like that. There's requests to go live. I didn't even see that. I'm sorry, you guys. Like trying to function, but I've really never done this before, so let's be real.
Hello.
The AI chromosome. Hello, what's up?
Nothing much, man. How about you? How are you doing?
I'm doing good. How are you doing this beautiful, I guess, morning? Is it morning over there still?
No, it's night in India. I'm going well, thanks. I just
So
I just heard that you were talking about plant tissue culture and thought I would like quickly pop in since I have a bit of experience in plant tissue culture as well.
You do? That's so
Yes.
cool. Wow. Go on. I'm so interested to hear.
So like as you rightly mentioned, plant tissue culture would, in your own words, would filter out the nasty stuff that is an unfortunate byproduct of our consumer-based economy. So if you use plant tissue culture and take a lot of explants for like plants, for essential plants like camphor, which is used in almost everyday products, but the packing and the processing actually um,
The oils, right? The
yep,
camphor oils that
camphor
you're
oils,
taking?
yeah. So it would have been like it would be more beneficial to the environment there using plant tissue culture. So
I totally agree. I mean, I think, and this is just a rough estimate, but okay, I'm going to walk with you real quick. If you take one plant, I mean, essentially just from this plant, how many cuttings can we make from that? Probably over 100. And
probably,
so
yep.
one plant, and I know that like you're saying, environmentally we will have a percent failure of like contamination, but we can go in the 30% range, and before you know it, one plant, if we were to get it down, like down packed aseptically, one plant would equal 1 to 150 plants. If we can do that with hemp, or there's a lot of mushrooms like mycelium out there that they're finding that is super sustainable, and it's also a spore, so it's more tolerable to conditions because it just lies dormant until the right conditions happen.
Applied, yep. We can use it for spores as well. Like we can use fungal spores which have been propagated naturally. So, yep.
It's, oh my gosh, this really makes my heart, it warms my heart to know that there's people all around the world that are thinking sustainable. I'm sure it sounds like you work in a lab also with tissue culture, and if you realize all of the paper waste and plastic waste
Yeah, we are a
Not
good contributor
initially.
to yep that yeah because
It's an,
generally yeah because it's like currently because we can't handle contamination, so we just pop it in and then pop it out, which is an unfortunate bycause that brings a lot of additional plastic waste to it. So
right, right. I totally agree. And I mean, I'm wondering, honestly, there should be a way to where all of us can join together. We should start like some sort of initiative or some sort of program which would catalyze this effort, because I've been saying for the last few years that we don't need any type of petroleum plastics. We should halt all production of petroleum fossil fuel based plastics. We have no use for it whatsoever. And
yeah,
you heard of these giant land masses, the islands of trash?
yep. Pacific Garbage Patch, and there's this another one on the west side as well. So
Right, it
yeah.
would have been like, I
I
think it was
was
the 12th
I was
largest
actually moved by the video in which the diver was literally driving like it was less of an ocean and more of the diver diving in a patch of I mean a pile of garbage which was dumped in the ocean. So
That's just insane. And I think that the fact that you and I, and I'm sure so many other people, see the vision of how the connection from tissue culture to sustainability, how that's kind of, it seems like such a obvious answer.
answer, yeah.
I think that this is such a good first step to find that this is like a collective from the Akashic records or something. This is on all of our minds. Like, we're all thinking this.
Yeah. Anyways, I'll
Yeah.
let you carry on with the stream forward and good luck with the stream. Thank you. It was nice talking to you. Have a nice day ahead. Yeah.
You too.
Yeah, brilliant. Bye.
Bye. Yes, hemp plastic. That is what we're talking about right now. We're talking about hemp plastic. We're talking about any type of sustainability that we can use through tissue culture, because it's so sustainable. Like, we're talking about just this plant alone right here, jade. I'm not sure about what the Latin name is, so please forgive me. But you can easily get over 100 cuttings, 100 plants, probably 150, equate 30% failure, and you are still going to end up with a large amount of plants. It's so sustainable. I'm all about it. Okay, Sammy, Ula, we're waiting for you now. There's a company out there making containers for the cannabis industry. Woo! Let's go. We're going to save the world.
Hello, how are you?
Welcome. Hi, how's it going?
Hello.
Uh-oh. Looks like... Hi. You're cutting out a little. There you are. Welcome. How's it going? Let's see. Oh, looks like they're having some problems, but I want to, yeah, I think that really talking about the sustainable applications, I saw somewhere where it says, oh, there's one company out there that's making hemp containers for the cannabis industry. And I understand that there's even biodegradable plastic that can be made from, I believe it's seaweed. So it's kind of like agarose plastic, which, if you will. But I think that is so cool. Tejas, Godbo, what do you think about stem cells? I think stem cells are cool. I think that using the T cells is going to be a really good idea. So when you're going into, we're talking about like with mammalian like cultures, correct? I don't know a lot about stem cells, but I did do a little bit of research in school about like, I guess like whether it is, I don't know, a moral approach on whether you're using like embryonic stem cells, but oh well. I think that it's pretty interesting, and I think that it's super important about everything. Let me see. Just joined. Do you study cannabis? I just caught the earlier Yes, so I have been a part of, in Los Angeles, I've been working with cannabis reform since 2011. So this means that I was there when you needed a rec and it was illegal. Like if you had a joint, it was super illegal to drive around Los Angeles, and it was scary. And I used to work at a collective that we were always worried if we were going to be raided. Like there'd be police staking us out down the street, and like here we are helping like a 80-year-old grandma, like because she has arthritis and wants to smoke a joint. But yeah, actually, more importantly, we've been there at the steps of City Hall in downtown LA protesting for cannabis rights for recreational use, for medicinal use. Right now, a really good friend of mine, Anthony Turner with BBC, he just did a interview. Go look it up, Anthony Turner, BBC. It's with LA Log Cabin, and it's on social injustice or social justice, and it's really about expunging people that have been in prison for nonviolent cannabis offenses. But also, more importantly, when we talk about the cannabis plant, I definitely think that using hemp and cannabis it's going to be one of the biggest steps we can take towards getting us out of this global crisis. Like, let's be real. There's an elephant in the room. We in trouble, people. We frickin' in trouble. We all know this. So let me try and get this person back on. Waiting for Sammy Ula Katak. And I think that that's probably we all know this, but that's one of the reasons why cannabis and hemp was illegal. And
Hello?
Colorado legalized psilocybin for those that are out here. Welcome back. How are you doing? Oh, I see it. It's loading. It's trying. So to bridge the connection for everything, tissue culture, bunch of different media, super flexible when it comes to the different types of plants you are seeking to. Yes, it's in a lab. Yes, it's aseptically, so it's actually going to use a lot of plastics and resources. But the fact that we can use tissue culture to cultivate plants such as hemp that can be renewable and can make plastic, or there's even the seaweed that makes agarose that can also become biodegradable, and then linking it into social justice. This is a global issue. And this is actually boiling down, in my opinion, to taking on like large companies such as petroleum. So glad I can say that. Let me view, requested. Yes, let's go live. So I'm inviting someone else. Rathod. Sammy, I'm so sorry. I kept cutting out. Hello, are you there? Welcome, welcome. Oh, it was a network problem? Damn. I wish you can come on. I'm going to try and get... Oh, I can't say your name. I'm so sorry. Unjinjo. Super sweet name, though. DuPont has a shit ton of money. DuPont. I got to look them up. DuPont. Is they evil? You think they're part of the elite? I'm just playing. Hi! Hi, welcome everyone. We're talking about tissue culture. We're also talking about taking on the world, which means taking on the man, which means being sustainable, saying no to fossil fuels and petroleum, and just living a sustainable lifestyle. Like talking about tissue culture as being one of the most sustainable, I think. And why? That is because you can, any plant, if you fall a tree, you can tissue culture it and continue. Yeah, they really are part of the lead. Pretty scary. Yeah, we should talk
Hello,
1710 WV. Hi, welcome. You made it.
hello,
Hi. How's it going? Um,
hello, man.
hi. Do you have any questions about tissue culture?
Yes, I have one problem, and I'm I'm doing BS and bioinformatics.
Okay. And go on.
And I have one big problem. I'm doing research and PPCK2 genes and Arabidopsis thailand. Then I don't understand what I do and this genes. Please,
Oh, I think you're cutting out. So, are you trying to breed for a specific genetic trait?
phylogenetic tree, yes.
Okay, and so are you working in an arboretum, or how are you doing this? Like, is there you're having them like pollinate? Are you doing manual pollination, or how are you having them? I don't
Yeah,
know.
I work in Arabidopsis thailand plant, and sir give
So I'm wondering, what is the genetic? Are you trying to breed for a larger yield? Um, is there like a disease mainly that you're running into? Because also diseases, I'm
uh,
also really big
how
on.
I will find its C3 or C4 level. This 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 gene involved in photosynthesis process.
The gene is. I'm sorry, the video is kind of cutting it.
So remember net problem.
Yeah. Gosh, I wish I could help you answer it a little bit. Hey, Arashi, what's up? Yeah, I think, is this a type of mold that, like, maybe a disease that
Can,
your
can you
plants can?
use WhatsApp?
Hmm? Okay, I'm gonna end it just because it's not
You
the
use
network.
WhatsApp.
Um, for, are you talking about to make it so it's like aseptic? I use I think it's a bleach solution that you can use. That's 15% bleach solution. So if you want to make that, and you can have it soaking your cuttings, is that what you're running into? Probably contamination. I used to be ashamed of the pollution I created in the last few years, but I plan to fix that with cannabis. Nice. Yes. See, we can make everything that we need.
Hello.
Yeah, I totally agree. I'm gonna close,
Mm.
the video out just because it's a little laggy. Yeah, I think that for 710 WV, tissue culture is so important. It's basically in alignment with vertical farming. So it's using like a really small amount of land. Let me just be blunt. I'm really mad at the agriculture system right now. One of my biggest passions is agriculture too. So it's like, how the hell do you love agriculture, but you're mad at industrial agriculture? Yeah, I'm pissed. I think that industrial agriculture is awful. The nitrates, like nitrogen, phosphorus, is being leached into our water tables along with antibiotics in mass quantities. Like it's kind of like our world is doing a freaking beer bong of nitrogen and phosphate and a bunch of nasty leaching. What's my name? My name is Elnura. Pleased to meet you. And so, yeah, it's really upsetting that the agriculture system is not recognizing tissue culture. Tissue culture is still kind of like a niche kind of thing. I mean, do you guys agree? There's not hella tissue culture going on. I mean, I don't have a huge global awareness of this, which I'm working on improving, but yeah, I came from industrial ag and converting. That's so important. Sustainable agriculture. So my philosophy is basically two sides of the coin. There's conservation ecology, which is basically the fauna, flora, bacteria, soil health, everything in that sense. We want to maintain a biodiversity. So, like, for example, the monarchs and the milkweed, super huge trouble. It's not because of logging and agriculture that everyone thought, well, it is, but it isn't. What it mainly is the plant is being destroyed. So we are ending up not having a large loss of this super important pollination balance that's been going on for years. I think that there's like the population literally went down by like 10,000 since like 2014 from the U.S. Forest Service survey. So it's just really crazy that that's the way it is. Yeah, pollution to the ecosystem, glyphosates. I've worked with people that have like old school landscape kind of folk, and they're like, oh yeah, glyphosates. Okay, first of all, it's a nicotinoid, so that's like the shit you're smoking, excuse me, that's what you're smoking in cigarettes. And then second off, they're going to say that actually Roundup claims that within, I think, 48 hours, glyphosate has totally become denatured and is no longer basically polluting our environment, which I want to call BS on, because I think has a higher half-life, if you will, than stays around, and it does cause a huge deficit to our ecosystem. Yeah, that's why they fail pesticides. That's also why you see like old timers, because I feel like I have a decent amount of friends that have been in landscape business or agriculture or horticulture since like they've been doing it for the last 25 to 30 years. And I always hear a story about a friend of theirs that used to be spraying glyphosate. They'd be spraying it like that was their job. They just walked around with a tank on their back and sprayed glyphosate all day. Yeah, that is basically those people are sick now. They're not doing good. They mysteriously got ill with some sort of chronic illness. And so I don't think that is somatic hybridization good? Somatic hybridization. Well, I don't know so much like somatic. I know that hybridization is natural. I mean, we were just talking about hybridization last night, and we were actually talking about cannabis. So do you remember when there was actually strain-specific cannabis in the world? Remember when that used to exist? Well, so now it's everything's hybridized, but a lot of people are mad about it, and basically that's just kind of the nature of plants, you know, it's kind of what they do. So the somatic hybridization is the forcing of two different plants to, you're kind of forcing them to go together. So it's like taking like a grapple, remember grapes and apples? Kind of weird. But I don't know if we should be forcing it. I mean, we're here on the fine lines. We just got to be conscious to realize that when you are playing with genetics, be really careful that you have backup. You know, there's a lot of seed libraries that they'll have like an old heirloom seed and they won't realize that they have it, and they might crossbreed it with something else and they never saved the seed. And so now that genetic information is gone. Like, it's gone. Nobody saved it. So I think it's really important. Like, yes, somatic hybridization could be kind of cool. Just be careful. Also, save the original genetic information. This is not my forte, but for the example of the white rhino, that they had, like, the last rhino was living in captivity and nobody ever saved its genetic information, and now the genetic pool of that species is completely wiped out. It's gone, obliterated. So yeah, no, I just think that somatic hybridization, any type of hybridization, it's taking a original, for example, heirloom, pure genetic seed and forcing it to interact with a whole new genetic makeup. So I'm kind of cool with that. I think it's crazy, but also don't lose your old information. Like, that shit's important, dude. You don't want to lose our genetics. Biodiversity is key. Let's get Drew over here. Freeze it. Nice. Yeah, I have a bunch of Asclepias. Tissue cultures practiced in which country? More in which country? I don't know, man. That's or woman, whoever, dude. That's a good question. I think that tissue culture has like a small global community. I don't think there's a lot of people practicing tissue culture. I know that I've talked tissue culture, I've heard of it, but not a lot of people are actually performing it. I want to somehow do like a class or something, maybe a webinar, if you guys would actually be down to join in again. I want to make it to where tissue culture is like a household name, so that you can tissue culture with yo kid, yo grandmama. You could do it with whoever. Like, that would be cool, because, you know, it'd be really cool to live in a world where you're like, I'm getting cold out here, where you're living in a world where you're like, hey, my tree is getting really old. It's not infected or anything, but it's just like, it might fall over soon. I mean, let's be real. Oaks, quercus, those type of trees, they'll live a few 100 years, 200 years, 300 years, and then they just drop over. they fall over, and it's not from sudden oak death, it's just the nature of oak trees. Before it falls over and dies, cut it up, tissue culture. I think that's really cool. I think that that's really important as one of the ways to sustainably save our planet. There's like a government thing where they started tissue culturing every tree or plant that was healthy and would start regenerating it, and they had like a nursery or something. That'd be sick. Like we'd be able to save so many plants. Yeah. Hi, Bubbas. I've been taking clones of plants that you like. That's super cool. Cloning is really cool. I used to do a bunch of cloning. Just be careful if you're using rooting hormone. Don't get it on you. That happened to me one time, and I got like an infection or staph or some weird shit on my arm. I think it also had to do with using the rooting hormone, so just be careful. And I was wearing gloves, so I was also crawling on my belly through rows of plants. But anyways, I heard also that cloning, it's not a mother plant, as you know, so that's going to be another thing that the genetics just won't be as strong. Hi, Mommy. Hi, babe. That's my son. But yeah, I think that tissue culture is really cool because it's taking a part of the plant to where it's where growth comes from. So when you make the cuttings and the internodes, which is what we were talking about internodally, when you make a cutting, so between each of these little leaves, you're going to have a cutting. Doop, doop, doop. So between each of these leaves, that's internodally. You would take these larger leaves off. And that's actually where the plant has a lot of genetic information, and it takes up a lot of nutrients and everything. And so that's like, genetically, you're going to have more closer to growing from seed or having a mother. Is tissue culture good for ecology? Did you just start in this talk? Maybe. Tissue culture is key to ecology, in my opinion. It's super sustainable. From seed, right now, seed is really cool, but the thing about growing from seed is there's usually a higher percent error that you're going to run into. So like when you grow from seed, when the saplings are really small, to die. When you make cuttings from a plant, and you still have to work aseptically, so you still have to really on point clean environment you're working in. Pretty much you have to, it's going to be already like strong, so you're going to have to harden off the plant, which means once we open it up from this tube, shout out test tube gardens again, once you open up this tube, yeah, seed will age well as well. Yeah, that's true. Once you open up this tube, the air is going to burn the new cells. And so it basically, we need to let it slowly air out. And so I would poke a hole in the top of this lid. Dink. Where is it? Yep. I would poke a hole in the top of that, and I would let the air slowly start to get to it. Not the tissue. But then it will start to become more acclimated to the environment and the harshness of the air that it's in. But I like tissue culture a lot with cuttings. I mean, the seed is really good. Don't get me wrong, seed is badass. It's making a mother plant. But the cuttings, you already have a hardened off, like you have to re-harden it off, but you already have a really, hey, what's this discussion about? We're talking about plant tissue culture and actually how it can save the world and other sustainable important practices right now. So when you do the cutting, it's important because that cell has already been developed, like the tissue of the plant. We know it's already strong enough to sustain itself in the type of environment that it's been.